All right. So we'll move on now to our second speaker of the day, who is Swati Singh, also from IRIC, the Institute for Research on Immunology and Cancer at Université de Montréal. Swati is a PhD student in Dr. Smith's lab at IRIC that apparently uses a multidisciplinary approach that I'm very interested to hear about, integrating systems, structural, and cell biology to understand biological questions about the RAS oncoprotein and its alternate pathways. So same kind of setup as before relative to questions. If you have any, feel free to enter them in the chat. I'm going to do a better job of monitoring hands in the participants if you have any questions at any point. And Swati, go ahead, take it away.
Thank you for the introduction. Yeah, can you hear me?
Yes, I can. It's great. I was just, because I didn't hear you right away, I was worried that you were muted at that point, but no.
I was. Okay, thank you for the introduction and for the invitation. I am happy to be here, and talking about science is probably my most favorite thing to do. So hello everyone, my name is Swati Singh. I am a PhD student at Institute for Research in Immunology and Cancer at University of Montreal. This is our building, nice looking building at the campus. And right there, that's my lab. And a bit of introduction about me. I did my schooling and my graduation from India. I graduated in 2013. And after that, I worked as a research assistant in a lab where I realized I really love doing science and research. So I joined IRIC as a master's student, and then I transferred my program into PhD. And I am in my 4th year of PhD now. Soon I will be writing my thesis. And since I'm passionate about science and I would like to pursue a career in science, so I plan to do a postdoctorate after that, which would also take about 4 to 5 years. And that's required to be a principal investigator or a professor. Besides doing science, as Hilary had mentioned, I am also a member of the Student Association of IRIC, where we work towards advancing EDI at our institute and also bring about initiatives to improve the mental health of students. So what's my research about? I work on proteins. As most of you know here, that proteins are these cellular machines, or these large biomolecules that perform different cellular functions in the cell, such as it takes care of cell division, cell growth, it maintains cell shape, it takes care of the immune responses, it takes care of transporting different cargoes or vesicles around the cell. So if you consider cell as a factory, proteins are basically machines working inside them. So this is a pretty cool picture, which actually help visualize proteins as machine. This little guy here is a kinesin motor protein, and it's carrying a cargo around the microtubules, which are also protein polymers. So how proteins are made in a human cell? You might know the process is known as central dogma. It's a flow of information, flow of the genetic information from DNA to RNA, RNA to polypeptide chain. So the 3 nucleotide in the genetic information known as codon codes for an amino acids, and amino acids are the building blocks of proteins. So that's how a gene has now been translated into amino acids. So polypeptide chain, or a chain of amino acids, is the primary protein structure, which then forms these 2 structures known as beta sheets and alpha helices due to hydrogen bonding. And then they fold into these 3D structures known as tertiary protein structures. And finally, different amino acids can come together and form the final structure of a protein. So why I'm talking about the structure of a protein? Because the structure of a protein is really important for its function. So whatever function protein does, it depends on the structure that it has. Over the years, researchers have been able to solve and predict the structures of different proteins, and we can see that these proteins have different structures. That's why they are able to do different functions in the cell. Now, what's interesting is that any modification in the structure can lead to a misfunction. So a protein won't be able to perform its proper function if there is a change in the structure, which I will be talking about later in my presentation. So if that's how proteins are made in a human cell, how I work on proteins in the lab. So the knowledge that how proteins are made has now been translated in a lab setting. So I know how proteins are made in a human cell or in a bacteria. So we know what protein I am interested in. So I know the sequence of the protein or the gene of the protein. So I take that and put that in any of the system that's available to me. Most of the system are like bacteria, yeast, insect, or human cells. So we usually use bacteria and human cells in our lab. And these systems would make the protein that I'm interested in them. And then when I made that protein in a particular system, I rupture those cells and take out all the proteins from them. So now what I have is my protein of interest, plus the proteins that the bacteria or the human cell has made for its survival. So if you see this lane, I have like this bunch of proteins. What I do here now is some purification steps depending on the protein's charge, size, or other properties such as what it's binding with. So I use that information to purify my protein from the other proteins. And then eventually, as you can see here, I'm able to purify the protein that I'm interested in. So I have my protein now, how I visualize the structure of this protein. So there are multiple techniques and tools available to do that. X-ray crystallography, cryo-electron microscopy, and nuclear magnetic resonance are the three techniques that people usually use to solve the structure of a protein. In our lab, we use X-ray crystallography. So what we do, it's pretty cool. You make crystals of the protein that you're interested in, and then you shoot X-rays in them. The crystal of the protein will diffract the X-rays. What you get is the diffraction pattern. And then you translate this information from the diffraction pattern, which is actually coming from the electrons of the amino acids that are present in the protein, back to the structure of the protein. So you make the protein in a cell that you can't even visualize. And finally, you're able to visualize the protein that has been made in the cell. So I think it's pretty cool.
Swati?
So yeah.
There's a question from Halima. What's the point of purifying the protein? As in, why do we do it?
To study them. So I will be talking about that. I'm sorry, why did I stop sharing my slide? Okay. Yeah, to study them, because I will be talking about why proteins are important, the protein that I work on, and to study them, to do experiments on them, you will have to purify them to know how their structure looks like, and then finally know, okay, that's how they do their function. And if in a disease, something is wrong, you're able to understand why that protein is not functioning properly. I hope that answers your question.
I think that was pretty good. Yeah, thank you.
Yeah, and I think it will be clear in my slides further. Okay. So yeah, so we purify proteins and we do experiments on them to understand what are they doing in the cell and what goes wrong in a particular disease. I work on RAS. So RAS is it's a really important protein because it plays an important role in cancer, in causing cancer. So there are three isoforms of RAS, H and K RAS, and they are the most frequently mutated gene in cancer. They are present in about 27% of all human cancer, and KRAS mutation itself is present in about 98% of pancreatic cancer. So what kind of mutations are present in these protein are basically at these positions at 12, 13, 61. So I'm just going to explain how that happens. So we know that the mutation happened at the DNA level, right? So since I talked about central dogma, that the gene information is transformed into your amino acids. So if there is a mutation happening at the genetic code, it will be now translated in the amino acid sequence as well. And then I had said that any modification that happened in the amino acid sequence or the structure of the protein can lead to the point where protein doesn't do its function properly. And then any disease or anything wrong can happen in the cell. So here what's happening is cancer. So why a mutation in RAS leads to cancer? To understand that, we need to understand what does RAS do in a normal cell. So RAS is basically a protein which binds with these nucleotides called GDP or GTP, and it keeps cycling between these two states. It binds with GDP, and then with the help of other proteins, it removes GDP and binds with GTP.
Let's see. Hey, Chris, move around a bit. Oh, there, I see your eyes moving. Okay, so it's not me that froze.
Found with GTP.
Oh,
it's on. Yeah.
Well, so two things. First of all, you froze there for about 10 seconds. So if you could just back up 10 seconds. But also, while we're at it, there was a quick question here about whether mutation in BRCA, the BRCA protein, engenders pancreatic cancer.
BRCA protein? Okay.
Yeah.
I am not sure. They are in breast cancer for sure. They lead to breast cancers, but I don't know if they are present in pancreatic cancer as well. Yeah, I'm not sure about that. Sorry.
Great. Thanks. No, that's great. So yeah, if you could just back up by like 10 seconds where you were, because we've lost you just for a second there.
So as I was saying that it's important to know, so since mutations in RAS can cause cancer, it's important to know what function they do in a normal cell. So RAS basically switch between these two states. It's bind with GDP and GTP. What we know now is that when RAS is bind with this nucleotide, it's off, it doesn't do its function. And when it's bound with GTP, it's on and it does its function. What its function is that it acts as a switch to turn on different signaling in the cell. So it takes care of cell growth, it takes care of cell adhesion, meaning two cells coming together and further helping in the proliferation. It helps in the GTPase crosstalk, basically meaning it helps interacting with other protein so that the other protein can do its function. It's also take care of the cell size. So let's talk about how it takes care of cell growth, because that eventually can be helpful to understand how it causes cancer. So I talked about that RAS is cycling between these two states, off and on. So when it's on, it binds with this protein called RAF. And because of binding with RAS, now RAF gets a modification and it goes and binds with another protein called MEK. And now MEK has a modification done on it because it's binding with RAF. MEK goes and binds with another protein called ERK. And now ERK is modified because it's bound with MEK or activated. It goes inside the nucleus and it activates a number of transcription factors that lead to cell growth. So that's what is happening in a normal cell. What happened in the case of cancer when RAS has mutations in them, such as we talked about mutation at position 12 or 13. So research has shown that when there is a mutation in RAS, it cannot go back to its off state. So it keeps switching on to only on state. Now, when RAS is only on, the signaling that we talked about, it's always on. Because of that, your modified ERK goes in the nucleus and keep on doing the cell growth. So now you have aberrant cell growth, which is basically tumor or cancer. What's interesting is that, which is my project, what's interesting is that, and I did not talk about it, that RAS, although I said that it switch on so many signaling, it also switch on cell death in the cell, which is something that has not been studied a lot. It has not been paid attention because it causes tumor. So basically it's causing the switching on the cell growth pathway or the proliferation pathway. So cell death has not received so much attention. And that's my project on that. If RAS can lead to cell death in a normal cell, then why the oncogenic RAS is not leading to that? So that's what I'm studying in the cell. I don't have a lot of answers for that because that's going on, but that's about it. If you have any questions for this part, I would be happy to answer, or I would just continue after that.
Are there
Oh, go ahead, Chris. Shoot.
are there any theories? Are you going to talk about it? Because I'm
That's
very
what I was
curious.
going to ask too. I want to know.
It's this huge
yeah.
it's this huge paradox, because if the cell dies, cancer's over, problem solved. So
Yeah, I know. I think I did not include it because it just becomes so complicated. But yes, there are theories. So I am studying on these proteins which causes cell death, and what we know is that in cancer, the expression of those proteins goes down. So basically, cell is so smart that when it's this protein, this RAS is activated all the time, it can lead to cell growth. But since it can also lead to cell death, the proteins that can do cell death are also downregulated. So that's basically the theory that that's happening. But there are a bunch of different proteins which help in the cell death, and we are trying to understand if there is a way we could switch on the signaling of cell death for the therapeutic strategies. So that's my project on. Clearly, I don't have answers for that because the moment I have, I finish my pieces. But yeah, that's the theory. So a protein that I work on is called RASSF. It's a very similar looking protein that I talked earlier, that like this protein, it exactly looks like this, but it leads to cell death. And it's downregulated in cancers because of some modification that happened. So yeah, that's why it doesn't lead to cell death. Yeah. Okay.
maybe this is way too advanced. I mean, like, I'm what the question I'm about to ask. I mean, how could that possibly be downregulated? Is it more like there are more mutations that lead to them being downregulated? So you need a mutation in RAS and then a mutation in these other proteins that you're talking about?
So, so basically, I would say that cancer is not just mutation in one protein. It can be changes in different proteins, right? So, cancer is basically, we always talk about the oncogenic protein, the proteins that get overactivated and they do their functions like overgrowth and overproliferation. But cancer also happens when certain proteins that lead to cell death, when they are downregulated. So it's basically combinations of all these things. So what happened in the case of the protein that I am working on, the cell death, the modification happen when they are expressed. So it's called epigenetic modification. So they are basically after translation, something is modified in the sequence, and their expression level goes low, and they cannot do cell growth. So I would say that in cancer, it's not just the mutation of one protein, it's a combination of things that are happening. And then also because when RAS is mutated, since it's controlling so many signaling, we don't really understand what happens the first time. And then because of that, what basically changes along with it. So yeah, it's basically really complicated, but since we know that it causes cell death, can we switch on this signaling instead of this signaling, right? So that's my research topic is, yeah.
Thanks.
Also, I did not talk about the drugs for these proteins. It has been really difficult to find drugs for RAS, but now there are some drugs in the clinical trial. But since I would say that since we know that RAS is causing this signaling to be on all the time, there are drugs that inhibits the activity of this protein RAF and MEK, and they are used as therapeutic strategies for cancer, RAS-driven cancers. Okay, so
Swati, I have one more question for you here. Again, from Jonathan. For a benign tumor, does that work the same way in terms of the process that the benign tumor forms?
I wouldn't say so, because we know that mutations, like if the question is that for RAS, right? I wouldn't say so, because mutations in RAS are basically cannot be benign, because it really overgrows the cell and tumor happens, and it's oncogenic. It metastasizes really fast. So that really doesn't happen with the benign cancer. Yeah, these
Are
type of
benign
mutations.
tumors, are benign tumors still, if not directly from mutations in the RAS gene, are they still the results of malfunctioning cell cycles?
Yeah, to be honest, I'm not really sure if they are the result of malfunctioning cycle, but it is a malfunction of cell, because it is a tumor after all, and there is a tumor growth happening. That means your cell is not going to the cell death pathway. Yeah.
That's great. Thank you.
Okay, so I want to take a moment to talk about how it is to be working as a scientist. I would just say that what inspired me to be a scientist is science, because I get to do cool science, get to make discoveries, which can finally be contributed to cancer research or drug discoveries. But it's not that, like, it's not all good, because it's a long journey. I said that it takes around 4 to 5 years to finish a PhD. And then if I want to be in academia, I would have to do a postdoctorate. So there are a lot of challenges in the journey. My experiments fail sometimes my hypotheses don't work. So you just need to be persistent and keep doing it, because that's how science works. Sometimes things that you think that would happen in the cell doesn't really happen. But eventually I said that I want to end up in academia, but you obtain such a wide skill set that you can also work in biotech companies or pharmaceutical companies and help discover drugs for a lot of other diseases. And I would also like to talk about what I don't like about science. So as a person of color in science, I don't get to see people who look like me at executive positions or receiving awards that often. The representation of women in academia is skewed, even more for the racial or ethnic minority women. And the biggest example of that was seen in when the Nobel Prize was given this year in science, in these 3 categories: chemistry, physics, and medicine, to all men and no women. And this hasn't really happened for the first time. For of the more than 600 Nobel Prize given in the scientific discipline, only 23 have been given to women. So yeah, women are really underrepresented in science and in academia, but that doesn't take away the fact that there are amazing women who are doing important and wonderful work, and that has been translated to clinical trials and drug discoveries and a lot of things. That's why I think that initiatives like this are really important, where we celebrate women in science. So I would just like to thank you for organizing this event. And I would also like to thank the women that I work with in the lab who inspire me, and the women who, without being given enough recognition, just keep on doing science. And thank you so much. If you have any question, you can write to me. And also, if somebody want to visit the institute or our lab, we could make an arrangement. And thank you so much. I hope I've inspired you to do science and convinced you that proteins are cool. Thank you.
Well, you didn't have to convince me. I was already quite convinced about that. And, you know, I have to say, I personally really liked the beginning of your talk, and I certainly hope students from my class, and probably Chris's, appreciated it too, because you really hammered home a bunch of things that we've been trying to push hard in our class this semester as well. So I loved the link there. Matthew Smith looks like a kid. He looks like he's a grad student himself. Geez, he's
He's a new PI, and this is an old picture.
Oh, okay. Let's see. I don't see any questions right now. Does anybody have any questions for Swati?
I'm gonna speak again if you
No.
don't mind.
No, shoot.
I really wanted to express my appreciation that you also added the women of color, right? There's that extra challenge there that I'm always, well, sometimes reluctant to bring up, but I have discussed with some of my colleagues here at Vanya, and I think it's really important. I wanted to know, in terms of the science, and this question is maybe for Hillary as well, it does seem to me, and maybe you guys have numbers on this, that there's a bit more representation of women in the health sciences. Is that possible, and is there a reason for that?
Do you mean that there's a higher number of people in health science?
A higher number. It seems there's more women in health science than there would be in, let's say, maybe engineering or physics, certainly. And I wonder if anyone can speak to that, either you or Hillary. I don't know if it's completely off topic, but I remember speaking to some math teachers at McGill particularly, and they were really discouraged and aware of the huge drop off from one semester to the next in the number of women students in that, as opposed to a health science.
I am not sure, to be honest, I'm not sure about the numbers in health science compared to engineering or basic research. One reason that I think of, if I have to compare it with basic research, that it is a long journey and it's not easy for women. Like, if it takes around 5 years to do PhD and then postdoctorate, it's a long time. And of course, there are gonna be things that are not helpful if I am a woman to be in science. But in health science, you become a professional soon and you have a job. But the journey is so different for a scientist. I think it's just not easier to be a woman in academia. I think that is the reason, which, like, I think that is the reason. That's how I believe it, because I don't have a job. I am still a student. I'm around 30-year old, and I would be doing a postdoctorate. So that is not even considered as a job, like a profession. Neither it's considered as a student. So I am basically not a student, not an employee. And then I have to find jobs in academia where there would be some issues about it. So I think that can be a reason. And with engineering, you do see that the differences lies. And I'm sorry, Hillary.
No, continue. I cut you off.
I do see that how it can be difficult. So I don't know. I was trained as an engineer, and I switched to basic science because I did an internship during my engineering, and I really like doing basic research, and I realized that I would like to switch careers, and that's how I ended up doing basic research now. Yeah.
Thanks. I also wanted to echo Carl's thing. I had a few students who said, hey, we saw that in class. That activity really helped. Thanks, it was great.
Hillary, did you want to respond to that as well?
Oh, I mean, I completely agree with Swati. And I think when I think of health sciences, I also think of nursing. And I think that divide might go back to like decades and decades ago, when it was more encouraged for women to be nurses and men to be doctors, and especially even going back to the wars when men would go to fight and women would go into nursing. And so maybe that's just a perpetual kind of continuation of that. But I do agree. There's definitely like an imbalance, and I think it does come down to the length and money aspect too. And I think women eventually want to have kids, and that can really interfere if you're still a student for that long and starting a family and everything. So I think think the university is getting a lot better for accommodating that kind of stuff. And maybe that goes into answering the next question. Like I've seen a lot of scholarships recently for maternity scholarships, kind of for pregnant women or women who are giving birth to have scholarships and stuff. So I think that they're making an improvement, but of course there's a lot that needs to be done. I think maybe in terms of answering the next question, what do you think are the next steps for creating more space for women in the sciences, especially women of color? Can maybe be, more events like this and advertising events like this. And I'm not one to speak on that particularly, but I recognize the importance of it. And, just, I guess, I think speaking more about it as well and recognizing the discrepancies and shedding light on them and exposing them can make a big difference.
Thanks.
Yes, thank you. I'll speak from my position as a white male here, but at what was it? I guess it was a program committee meeting a few years ago, speaking with a representative from math from UQAM, who was a prof at UQAM, who was a white male at UQAM. He was talking about the challenge that they have in recruiting female faculty for their department, and how high quality female mathematicians are just they're snatched up right away, and how difficult it is, and how important it is for them to be able to show these female mathematicians in these positions as models that these younger girls can actually aspire to. And it wasn't something that I'd really ever thought of too much until he discussed this. And I've since come to realize the importance of that, and the importance of events kind of like this, like Hillary mentioned, that showing girls in Cégep how these women have achieved and where they've gone and what they're doing is a really good, excellent way to promote this and take these next steps towards improving female representation in science disciplines, in health sciences, but also in more applied sciences. And we've had some tremendous speakers on Tuesday. We had two from physics and from quantum computing, I guess. That were, wow, fantastic, really, really excellent. So I'm glad to be part of an event like this, because I hope that it inspires more young girls and more women to go into STEM related fields. Not specifically about women, though. I wanted to ask both of you, since you're studying at Université de Montréal, and maybe I'm being a little bit presumptuous here, but I'm gonna guess that you're going a little bit out of your comfort zone to end up at a French university in Quebec. And I don't really know what it's like in grad school there. And I'm guessing it might be a different environment than you might encounter as an undergrad. But for, since we're an English college, but where some students here might be thinking of pursuing or studies at a French university, I was wondering if you could talk about that, about your experience at Université de Montréal, and if you are going a little bit outside of your comfort zone, what you found the value was in that for you.
I think it's important to, study or choose a university based on multiple factors and whatever the one you think is best for you. When it comes down to language and the science, I know that English is very encouraged, so you could fully go to a French-speaking university and study science and have much success at that. But I think it is important because to keep English in mind. I mean, maybe I'm biased, but I know that it's spoken in my lab as well, with my lab having many French-speaking members, to be able to publish and write in English, because science is communicated internationally in English. So I think that is important to consider. But in terms of our experiences going to a French university, I think it was a really great experience. Like, I don't speak much French at all, so I think it was great. I mean, being able to go out of my comfort zone, and I've learned quite a bit being here, and I found it was nice because everyone was very encouraging for me to learn it and speak it. So ultimately it's been a very positive experience for me, and something I actually really encourage, like, no matter what you do, put yourself out of your comfort zone, essentially. I hope that kind of answered. I don't know about you, Swati.
Yeah, I think when I started, first of all, the thing is that since we are at the institute, we don't really go to the university. We don't have to deal with so much with the university classes because our classes happen at the institute. And since it's science, we all understand most of the science is done in English. Like we write our research articles in English, and most of the presentation that I attend at the institute are in English. So it hasn't been a problem for me. And my advisor, Matthew Smith, he speaks English. And so it hasn't been such a big, a drastic change for me. English is my second language. So for sure, I did think about it, but before joining the lab, I had spoken to Matt that I don't speak French, and I made sure that I did not take any French classes. I only took the classes which were taught in English. But definitely, if it has to be a master's or PhD, definitely it's doable if you don't speak French at University of Montreal. I also wanted to answer a few of the questions in the chat. This particular question, that what do you think are some of the next steps for creating more spaces? I think besides celebrating women in science and such initiative, I think it is high time we change some policies and make it easier for women to come in science, like in CGEP, making sure that there are women who are choosing science. Like as teacher, you recognize there is a problem, or maybe there is no problem, but I'm sure there is. And at the university level or at our institute, we need to recognize how many women are coming, pursuing PhD, and how many of them are actually ending up as scientists or having a career in science, because that really gets down. And you see so many women who want to be scientists, they don't really end up being scientists. So we need to recognize what's happening along that process and change the policies of the universities or the school systems to bring that change. And ask important question. I think Hillary had mentioned in her presentation, ask important question. I know it is difficult for me as a person of color, and I have faced problem, but I love doing science, and I would like to make this place more like more for the women who are like me, for more women, like easier. I want this place to be easier for women to do science. So I ask important question to the scientists or the leaders that we have in the institute. So ask questions. And the other question is, how you have the chance to be involved in science, why you choose it. It means what your motivation to dedicate yourself to science. I think I was always interested to do science as a kid, but I had mentioned that during your bachelor's, you can do internship at various institutes, and you can know if you like it or not, because it is a long journey and it can be frustrating. So find opportunities to gain the experience. You can also go now, like as a CGEP student, you can talk to PIs, like people who have lab running. You can talk to them, read about their work. And if you think you're interested in such work, you can email them and say them that, hey, can I drop by to the lab and see what do you do? Can I come and volunteer in your lab? And that would help you to know if you want to be involved in science or not, and if you want to pursue it as a career. My motivation, I just love doing science. I think lab is my happy place. So I don't really have an answer for that. It just tells me that I just have recognized that I love doing science, so I'm doing it. Yeah. And then you see it that as a girl, as a woman, you can change it. So change few things and can be part of the change. So that makes it more interesting. Okay.
Well, since you threw that in there, Chris, I'll throw it an auditory plug for our research methods course. It fits very nicely with segues very nicely from what Swati was just describing, that we have a science option course that's being offered next semester in research methods, where you get to, if you're part of the course, you get to spend a few weeks doing some research in an actual scientific lab. University, some things are done on Vanier, but a lot of them are off-site at other universities and actual research labs. And you do real research. It's a great opportunity to get your feet wet there, meet some people, learn about it. And Chris taught the course for a while. It's really, really a great course. If you're interested, just send me a Mayo and we can take it from there. Okay, 12:46. We're going to have to cut things short there because students have to go to another class. So this class period is over, but I want to thank both of you, Hillary and Swati, very, very, very much for your time today. This was really excellent. I'm sure everybody in attendance really appreciated hearing from you guys, hearing about your amazing research and about your experiences as women in science. It was really an excellent experience. Yes, that's what I'm talking about. Simantov, yes. So thank you very much to both of you. If you don't mind just sticking around for a few minutes, I'd like to say thank you to you guys in person. For everybody else, there's another one of these going on at 2:30 today. So maybe I'll see you guys then. Thanks very much.